going to go ahead and start on lesson 3.3, analyzing variation and reproduction. All right. By the end of today, we're going to test some predictions on which gene variation versions get passed from parents to offspring. I want to let you know, guys, we are going to start a new 9 weeks. I don't know if you heard from your other teachers, but we're going to start a new 9 weeks on March 8th. Since 6th grade is coming back into the building, we think it'd just be easier to start that new 9 weeks in. And so we're going to finish this unit next week. So you guys are going to actually have your test next Thursday. We're going to have a review on Tuesday, and then you guys will have your test on Thursday. So get ready for that. The first thing that we're going to do is our warm-up. And you guys are going to go into your Google Doc. You're going to explain why offspring have 2 copies of each gene. And you will see in the Google Doc that you need to make sure you use the words in the word bank. If you don't use all of those words in that word bank, make sure you're using at least 2 of them. But try to use all of them. All right, so use that diagram up there to help you explain why offspring receive 2 copies of each gene. And then be ready to answer in the chat. I'm going to start the 2-minute timer. Y'all go ahead and do that warm up in your Google Doc. The vocab words. Let's look. What are the other ones? Genes, inherit, offspring, and parent. So Momini has used all of them. They may get the same copy from creating an offspring, they inherit a copy of the parents' genes, one from mom and one from dad, so they get 2 copies of the genes. So Kendall's explaining the picture really well because it's showing that you get one of those copies from your mom and one of those copies from your dad, and that is the reason why you have 2 copies. Right? Remember, we got rid of that claim 2 the other day, and we know that we get 1 copy of a gene from our mom and 1 from our dad, and so that is why we have 2. Alright, so the next thing that's going to take up most of class is our sim. So we haven't really made very many predictions with the sim. We've only been filling out the table or looking at what the 2 spiders produce for their offspring and what their traits and gene versions are. And so what we're going to do today is we are going to predict some of those gene versions, and then we're going to test the possible traits of spider offspring. Alright, so we're going to investigate this question. How does sexual reproduction result in variation among offspring? Alright, how does it result in variation among offspring? So now that we know how organisms inherit their genes from their parents, we'll look at why spider offspring have different traits for self-flexibility, even though they have the same parents. So we're going to make some spiders today and predict the gene versions, and we'll use this sim to test their predictions. So the first thing that we're going to do is I know that the Google Doc looks like a lot, but the reason why it's 4 pages is because it's spread out, and there's quite a few tables that we're going to use on there today. And so the first thing that we're going to do is make Greg and Ann in the simulation and make some predictions. Right, so we're actually going to do that together. And then I will let you guys go on your own. So this first table, the one that's on page 1, this first table and the highlighted in yellow words are what we're going to be looking at first. Okay, so let's make some predictions. We're going to launch the sim. Make sure you're pressing that sim, and I'm going to split my screen into 2 so you can see my sim and you can see my Google Doc. Alright, so you should have your sim open, unless you just want to look at my sim, but at least you should have your Google Doc open. Okay, we're going to do this first part together, and then if you scroll down a little bit, you'll see kind of the same thing, but the blue table, and then scroll down a little bit more, and you'll see where you have to test your predictions. That's what's going to be next. Alright, so for this first part, we are looking at Greg and Ann. Alright, we're looking at Greg and Ann. So what we're going to do first is we're going to mate Greg and Ann. Okay, we're looking at silk stickiness, so I'm going to go ahead and pick that feature. And let's take a look at their trait and their gene version. Somebody tell me what the gene version for Greg is and his trait. Alright, so his gene version is S1, S2. Everybody's telling me in the chat what is his trait for silk stickiness. Right, it is sticky. Now, what about Ann? Good, it's the same thing, right? So we can put S1, S2, and then she is also has the the heat trait for silk stickiness. Alright, so what I've done on here, we've seen kind of the same version of this table, but in this case we're making some predictions and not just getting them and pulling them from the offspring that we see when we mate these two spiders. Okay, so I'm going to leave it on this screen for right now, and then I will create reproductive cells and fertilize them so that we see the actual offspring. Okay, so this is just the predictions. This is not, we're not pulling this from the offspring that we get. Okay, so the first thing that we're going to do, this is almost like a Punnett square. This is Amplify's version of a Punnett square. Next week, we're actually going to do, not next week, but the week after, before you guys come back to school, we're going to do kind of a project-based learning thing, and we're going to learn more about Punnett squares, which I think will be more interesting. So this is kind of like our own Punnett square. And then below here, I've actually made just a short little table. It's not on your Google Doc. And we're going to do a Punnett square so that you can see how both of these relate. Alright, so we know that we can have these 4 different gene combinations. Right? We get one from our parent, we get one gene from our parent, and one gene from our mom. So right here in this first column, my first possible gene combination, I'm going to pull that S1 from Greg, and then I'm going to pull the S1 from Anne. You guys see right here, I actually put the S1, S2 up above here so that I remembered what I'm working with and what the two parents had. I just put it down here, but it is up here, and you can see it on the stem. So if they get S1, S1 from both parents, their gene version is obviously going to be S1, S1. You just push them together, and it is their gene version. Alright, now I'm going to stay with S1 from Greg. But now since we've used that S1 from Anne, we haven't used the S2 from Anne. So that's what I'm going to use from Anne. And so when you put those together, it is S1, S2. Alright, then we've used S1 from Greg 2 times. We've made as many combinations as we could with Greg having S1 and then Anne's S1 and S2. So we're going to move on to Greg's S2. So that part of his gene version. And then to create another combination, we're going to go with S2, S1. Here's where you may get confused, just a tad bit, because when I write this is the same thing as saying this S2, S1 is the same thing as saying S1, S2. If you want to write it S1, S2, I think it will be not as complicated because you know it's the same thing as this other gene combination. So technically here we only have 3 gene combinations because S1, S2, and S2, S1 are the same thing. They're just flipped around. Alright, and then the last one, I'm gonna pull that S2, S2 from both of the parents, and so I would eventually get S2, S2. Alright, so down here at the bottom, I'm going to make my own Punnett square. Alright, so we'll talk more about recessive and dominant. You guys are, I feel like, pretty aware. Most of you guys are pretty aware of what dominant and recessive might be. But we're going to learn more about that, and not next week, but the week after when we do some Punnett squares. And so I think looking at this Punnett square gives us a good idea of why we just wrote what we wrote in that table. Okay, so this is how I would set up my Punnett square. I got S1, S2 at the top for Greg, and S1, S2 on the side for Anne. And so what I do here is I'm just going to pull down what I have at the top and pull over what I have at the side. Right, so I would have S1, S2, S1, S2, S2, S2. Right, so I'm pulling down what I have at the top, pulling over what I have at the side. It's just going along with what's in each column and what's in each row. Now, if you look at this right here, what I have highlighted is the exact same thing that I have over here for the offspring's gene version. All the possibilities for the offspring's gene version. Alright, that's why I said it's so similar to a Punnett square. It's just kind of it's a little more than what you would have in an actual Punnett square. Right? So I'm going to go in, I'm going to create reproductive cells. I told you all last class about this next step right here where I wish it would freeze when it got to this, but it doesn't, so we'll just have to kind of get over it. Now look at my offspring. What do you guys notice? What do we notice about what's happening in the sim that I just did? Point out anything that you notice. Okay, so now this says the spider with S2, S2 have no sticky silk. So maybe that's when I talk about recessive and dominant, maybe that's not recessive, sorry. Yes, I'm getting myself confused. When I say dominant and recessive, that might be a recessive trait. So for a spider to be sticky, it might be dominant. For a spider to be not sticky, it might be recessive. Lacey, you say they're all yellow. Okay, that's something good to point out if we were talking about body color, because Anne is colorless. All right, do we see all of the possible combinations for the gene versions? Right, Lacey said we do not. Reagan said we don't see them. We don't see that S1, S1, right? And my guess is that S1, S1 means it might be sticky because the S2, S2 says it's not sticky. Right? So the next thing you guys are going to do on your own is that next part of this sim. So if I scroll down here a little bit, you're going to make two different spiders. Right? So you should be mating Otis and Ruby. So on mine, it's different. I'm going to scroll down on your all's Google Doc. You guys should be mating Otis and Ruby. So if I go back into where you first get to the spiders, you should see Otis and Ruby down here at the bottom right. Okay, so make sure you're mating Otis and Ruby. And before you get into the body color feature, you're going to point out Otis's and Ruby's gene version and trait for body color, and then you will predict the different gene combinations here, just like we did up top. All right, I'm going to allow probably about 4 minutes for that. Okay, I'm going to give you guys a 4-minute timer. Point out Otis and Ruby's gene version and body color trait, and then fill out that table there for the different gene combinations for the offspring. And I will scroll up pasted together so that you can see how that's supposed to look. Alright, make sure you are done with that. If you would like to move on to testing your predictions, you can, but I'm going to allow for some more time to do that, and I'm going to explain that table real quick after you guys get done with this. Alright, what were the different gene versions that you guys got from Otis and Rudy and their body color? So what do we put in this blue table right here for the last column? Alright, so one of them could be C3, C2. Alright, so Kendall gave me one, so I'm going to give you another one. What's another possible gene version for an offspring? C1, C2, C1, C3, I'm gonna write these down so I don't forget them. And the last one is C3, C3. Good. Thank you guys for responding in the chat. So these should be your possible combinations. They may be in a different order, but that is just how I put them here. Alright, so we got 4 different possible gene combinations because their gene versions, they have all of the different gene versions. So C1, C2, and C3 are present. Okay, so when I create reproductive cells, this will be your next step when you test your predictions. Randomly fertilize. This should be what you put in your next 2 charts. Okay, so when you do test your predictions, you'll use this first simulation to write out all of the gene combinations from each offspring. So offspring 1, 2, 3, and 4, you'll use for spider 1, 2, 3, and 4. And all you're doing is just putting that gene combination or gene version in that column. So my first spider is C1, C2, and the trait is brown. So that's all I'm putting in this table. Alright, in order to test it twice, all you need to do is click this back button up here in the simulation and then randomly fertilize again, and you will get a totally different set of 4 offspring. Alright, I'm going to give you guys about 3 minutes to do that because that shouldn't take you as long to test this out. So I'm going to set a 3-minute timer. You all can start. Kendall says one of her spiders was colorless. I keep trying to go back and randomly fertilize again to see if that happens. Alright, so in order to be colorless, you have to have that C3C3 combination, right, for your gene version. And so if you're looking at this table right here, you have a 25% chance of being colorless. And when I say 25% chance, the reason why I say 25% chance is because there are three other gene combinations that you could have. And so 1 out of 4 would be 25%. Right? And so you are more likely to probably either be yellow or brown. Right? So make sure you fill out that chart for testing your predictions, and we're going to move on to our next thing. Your activity 4, that homework, will be the last thing you need to do before you submit your Google Doc. So you guys will have time either at the end of class or right after to finish that homework, because it is not very much. You're just responding to a statement and whether you agree or disagree, and we've done that many times before. Alright, so the next thing that we're going to move on to is the modeling tool in Canvas. Okay, so we're actually going to be looking at Venom. So the feature for Venom. You guys can go ahead and go into modules in Canvas. Take a break from that Google Doc. Make sure you go into modules. And you will see Activity 3, Modeling Venom Inheritance. Okay, Activity 3, Modeling Venom Inheritance. And so I'm going to pull up my copy of it. And you should see something that looks like this. I'm going to go through it, I'm going to explain it, and then give you guys some time to finish it and turn it in. Okay, so while y'all are pulling it up, I'm going to have it on my screen, and I'm going to go through the goal and the instructions. So your goal is to use the Venom as an example, to show how the gene versions of siblings can be different from each other and from parents. Alright, so your instructions. Step 1, what are the 3 possible allele combinations for the offspring? You're going to place the alleles, which is V1 and V2 in this case, for each offspring over the yellow circles for the 2 possible alleles inherited from the parents. And so when I say alleles, I'm only talking about 1. Alright? And when I say 1, I mean the V1 or the V2. When I put the V1 or V2 together, that creates a gene version. Alright, so when I say alleles or when I say gene versions, I'm talking about the same thing. Alright, remember that. When I say alleles and gene versions, I'm talking about the same thing. Alright, so the different combinations that you could possibly have from these parents. What is 1? Somebody tell me, what is 1 possible gene combination that you can get from this parent? Well, 2 parents, obviously. Alright, Kendall says V1, V1. So that's what I'm going to go ahead and do, is drag one of these over here, and you're going to put it in the yellow circles. So I'm going to go ahead and drag V1, V1, and put it in these yellow circles. Now, the next thing, your test is going to talk a lot about the certain kind of shapes of proteins and how they fit together. If you look at these proteins, we're going to talk a little bit more next week because we haven't talked too much about how they actually fit together. We know that our genes give our ribosomes the instructions to make these proteins, and proteins make a certain type of trait. but we haven't talked about how they fit together. We're going to talk a little bit more about that next week because that is important on your test. But in this case, we're trying to decide what protein goes into the cell. And so I've told you guys many times, when you're looking at this, you have V1 and V2 in this case. There is, if you look at the simulation, a V3. That's why this protein 3 is right here. But what you want to decide is what protein goes inside the cell if you have the gene version V1, V1. All right, so if you need to look at your simulation and tell me what protein needs to go in the cell, you can do that. But if you remember what I said, when I say V1, it also lines up with the first protein. When I say V2, it lines up with the second protein. When I say V3, which we don't have, obviously, because neither of the parents have V3, that would line up with the third protein. All right, so in this case, if I only have V1, V1 for my gene version, what protein is going to go in my cell? Correct. Get those correct, we're going to put protein 1. So all you need to do is drag protein 1 over and put it into the cell, the blue circle. All right, now the last step, step 3, drag the words below to the pink boxes to indicate the genotype. All right, so it's either going to be homozygous or heterozygous. If my gene version is V1, V1, am I homozygous or am I heterozygous? All right, homozygous. Lacey, Kendall, Natalie all said that. So what I'm going to do is I'll just drag homozygous over and put it into that pink box. And then you guys will figure out those next 2 possible offspring gene versions and do the other 2 steps. All right, I'm going to give you guys another 5 minutes to do that, and then we will wrap things up. Okay? So make sure you are working on the modeling tool and Canvas, and I'm going to set a timer for 5 minutes. Hopefully it won't take you guys that long, but you have that time to do that. If you want to move on to the homework after this modeling tool, you can do that.
When school isn't a place you have to be, why not go to school where remote learning can be as remote as you want?
up this lesson. Okay, so remember, we're investigating variation within the spider family. So the spider offspring have different traits from each other, and some even have different traits from their parents. Okay, so we got rid of claim 2 last class. Alright, so I want to know what you guys are thinking right now in regards to the claims. So are you thinking claim 1 or are you thinking claim 3? Because next week on Tuesday for you guys, we will support on those claims and start writing just a little bit before we review. And so Jack's school claim you are leaning towards. I'm going to zoom in just a little bit so you can see those claims a little better. Alright, so the claim 1 is talking about mutations. Affecting the traits, and claim 3 is talking about offspring inherit different combinations of gene versions from their parents. Alright, so saying claim 3, when we go around here, kind of on the fence of claim 3, claim 1, that's totally fine. But on Tuesday, we will be writing just a little bit, just a short kind of thing to wrap up everything that we learned. Alright, but we have key concepts here. Through sexual reproduction, each offspring can inherit a different combination of gene versions. Therefore, siblings can have different traits from each other and even from their parents. So when I'm talking about this, I'm talking about maybe you are a redhead and maybe your parents have dark hair or they have long hair and you got the gene, they both had a gene that coded for red hair and you got that gene. So when you guys saw things in that simulation, like one spider, the dad had brown color and mom was yellow, and one of their offspring was colorless, that's the case of that because they're both carriers, but they don't have the actual gene. Just make sure you just write down the only thing that you need to put in your digital notebook for me today. Alright. Make sure you have keep all this down and keep that up just because you may be still typing. The last two things I want to think about, if you wrote on the homework for the Google Doc, make sure you do that. And then you have a weekly check-in if you have not done that already. And that will be due on Sunday. And make sure you finish up that Google Doc and do the homework, turn that in, and then you have your weekly check-in. Alright, if you guys don't have any questions, you're free to go. If not, just ask, and I will see you guys tomorrow.